Welcome everyone to the Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. The session is currently being recorded and it will be posted later on our website. At the end, there will be a Q&A session, so please add your questions in the Q&A feature below so that we can moderate the questions. And now, Dr. Mustelin, I'll let you introduce our speaker for today.
Well, thank you, Danielle. It is a great pleasure and honor to welcome today's Grand Round speaker, Professor Kenneth Smith. So Ken got his basic training, education, and medical degree in his native Australia from the University of Melbourne in the 80s. He worked as a physician and internist and eventually nephrologist in Australia for a good number of years until he enrolled in a PhD program and got the PhD degree in 1996 from the Weihai Institute, studying B-cell apoptosis and antigen production, antibody production. With his fresh PhD, he relocated to the University of Cambridge, where he worked for a very good number of years in increasing academic rank. In 2009, he became a full professor of the University of Cambridge. And in the following year, 2010, he became the head of the Department of Medicine, a position that he still holds. And then in 2018, he also became the director of the Cambridge Institute for Therapeutic Immunology and Infectious Disease. And I think that characterizes Ken's work over the years very much. He has done an enormous number and breadth of investigations in understanding the immune system in human disease, starting with the B-cell angle and understanding the differentiation of B cells into plasma cells and the role of cell death, the role of T cell help in the lymph node, including the regulatory T cells that look like follicular helper cells and participate in controlling the B cell response. He has worked in a good number of rheumatological conditions. One of his focus area has been vasculitis, where he's founded the European Vasculitis Genetics Consortium. And he's investigated T cells and the genetic basis of disease, including now in the Cambridge Institute for Therapeutic Immunology, clinical trials with a variety of therapeutic approaches. I've known Ken since 2012 when he served on the scientific advisory board at MedImmune during my time there, and I really came to appreciate his insight and wisdom and major contributions, including a very interesting paper that came out at that time from his group in Nature that concerns T cell exhaustion, which we know in immuno-oncology as a problem because it prevents T cells from eradicating the tumor. But it turns out in his work that T cell exhaustion is a good thing in patients with autoimmune disease because it gives a improved long-term outcome where the immune system decides not to be so activated and to enter the state of exhaustion instead. So I will look forward to your talk, Ken, and the title you can all see on the screen, and I will hand the virtual microphone over to you, Ken, please.
Thomas, thank you very much. Thanks for your very kind invitation and for your description of my work, which reminds me of quite how unfocused I've been over the years. I also apologize for the fairly prosaic title, but what I'm going to talk about today is not the exhaustion-related work that we've been doing. That will have a terrific update, I think, but not till next year. But to focus on some work moving between an understanding of genetics and disease mechanism. That is the culmination of some work that's taken us a few years. I should say I'm not a rheumatologist. I'm a nephrologist by training. I still do general medicine, unfortunately, for my sins. So my excitement from the clinic from last week was I was asked if a patient had TTP, thought that she didn't, and I was right, because she ended up with Lassa fever. It's the first case reported in the UK since 2009. Unfortunately, one of her family members then died of it. So, but I know nothing about Lassa fever, I should add. I'm not going to talk about that. I'm going to talk about primary immune deficiency, but I'm going to talk about it as a route to understanding more common autoimmune diseases rather than as an end in itself. And so most of you will be aware of primary immune deficiency, at least on the periphery of your experience. It's a group of conditions which are lumped together and defined primarily by infection susceptibility. So you don't really have primary immune deficiency unless you have recurrent or severe infectious disease. But about a third of people with primary immune deficiencies develop some sort of autoimmunity. About 10% develop malignancy, usually, but not always, lymphoid malignancy. And atopy is also quite common. And this is an area of some interest because there are clear benefits to defining the genetic underpinnings for patients with primary immune deficiency, because they can benefit from genetic counseling. You need a diagnosis to be eligible for bone marrow transplant. And increasingly, gene-specific therapies are becoming available, perhaps even one day more widespread gene therapy. But most primary immune deficiencies don't obtain a genetic diagnosis. And the reason for that is shown on this rather amorphous slide. And that's because while we're all aware of the children with severe primary immune deficiency, kids with SCID who end up with early bone marrow transplantation, most people with primary immune deficiency are in fact adults. And most of them have sporadic disease, have no obvious family history. And so that makes it actually very difficult to use traditional genetic approaches to reaching a diagnosis and finding new genes. A subset of them, between 10 and 20% of adults with primary immune deficiency do have familial disease, and in that subset, monogenic discovery is possible. So we tried to work out to determine if we could make some advances in understanding the genetics of this large group of people. And I should say they're a difficult group. The reason they're difficult is because they're adults. Penetration in the family is a clinical penetration of disease is very hard to assess, because of course, just because you don't have disease at 30 doesn't mean you aren't going to get it when you're 50. So because it's age related and these are adults, it's hard to assess families in that sense. The other problem is, of course, it's hard to diagnose this disease. What one person, if you present with a second episode of pneumonia, some doctors will say that's bad luck. Others will wonder if you've got primary immune deficiency and test you further. So I think there's a lot of blurriness between the normal population and people with immune deficiency at a diagnostic level. There's also blurriness at a clinical level. If you happen to get the autoimmune manifestations of immune deficiency first, you'll end up in a rheumatology clinic. You'll be most likely given a different diagnosis, and then whether or not the underlying immune dysregulation syndrome you have ever comes to light is not clear, because of course your subsequent course will be impacted by immunosuppression. And if I think about the vasculitis clinic, for example, that I work in, that David Jane runs here in Cambridge, it seems to David over the years that there is a pretty high frequency of infection among patients with the autoimmune diseases that he sees. And of course, we tend to put that down to the immunosuppressive therapy, but perhaps it's down to the primary immune dysregulation that drives both the autoimmunity and perhaps a degree of immunodeficiency. So how do we understand this? It's going to be complex. It's not going to be simple Mendelian recessive disease like in kids with SCID. So we approach this by becoming part of the 100,000 Genomes project in the UK, which was developing cohorts of whole genome sequenced patients of different types. And these are all the different projects. We were the second biggest of the two. The biggest was the bleeding and thrombotic platelet disorders group. And this was a nationwide sort of series of cohorts. And ours was put together by James Theventheren, who was then a postdoc. He's a clinical immunologist, was a postdoc in the lab. He now runs his own lab in the MRC toxicology unit. So James pulled together this group of, I think it's 27 of the main immune deficiency treatment centers across the country. We had a series of very dedicated clinicians who ran MDTs and helped feed our results back to the referring clinicians, and then a team of people who contributed more to the research, and particularly these guys who generated the data I'll show you. And so the cohort now stretches out to about 1,400 patients that we've whole genome sequenced, and that's about a third of adult patients with primary immune deficiency in the country. And we've got funding to double that number. And this is their clinical manifestation. So most of them have CVID. Some have of the forms of common adult immune deficiency. And there's relatively few that have autoimmunity only, plus in vitro or phenotypic, immune phenotypic abnormalities. And of course, very few have SCID. I'll talk about one of those later. But the thing to note about these is that the great majority of them in blue are sporadic cases without family history. And as you'd expect, this population, they all have recurrent infections. About a third of this first 730 or so that we analysed, in some cases, I'll show you the data from just over 800. A third had autoimmunity, some had defects in T cells, and about 10% had malignancy. So what we did was to get this cohort, as I've mentioned, predominantly adult and predominantly sporadic. And the first thing we did was to look for known genes, the 300 or so, and there's now actually 400, but when we first did this analysis, about 300 known genes that cause primary immune deficiency. And then we used a Bayesian approach to look for new genes, and then started to explore things like the non-coding space and the role of common variants. I'll take you through the first bit of this relatively quickly, because what I want to focus on is how common variants can inform our understanding of rare disease. So firstly, when we looked at known genes, we found a diagnostic yield, which is sort of what we expected. About 170 of these 800 or so patients contained a predicted damaging defect in a gene known to cause primary immune deficiency. If the mutation itself was previously reported, it's in green. If it's a novel variant, it's in orange. And you can see lots of well-known immune genes along the bottom here. So this is sort of what we expected. What we didn't expect was that in those syndromes where there's we think there's a relatively well-defined clinical phenotype for these patients, in all of the cases in pink, the clinical phenotype was wrong. And so what we found was that, in fact, there's far less clinical homogeneity within these syndromes than has been reported. The reason that that's the case, we think, is that what tends to happen in these genes are first reported as being associated with primary immune deficiency in a handful of cases. 2 or 3 families perhaps might be the first report. And what tends to happen is a clinician finds a patient with a certain phenotype with a variant, and then looks for all the other patients with a similar phenotype to find the next variants. And there's a sort of self-fulfilling prophecy that the first report has a sort of finite phenotype, but there's much more variation than we'd expected. But what we then did was to see if we could find rare variants from this really cohort of index cases, of individual cases. And to do that, we used a method, or Daniel Green and Ernest Turry used a method that they developed, which was taking a Bayesian approach to identifying potentially new immunodeficiency causing genes in a cohort like this. So what they did was to essentially factor in things like whether the mutation was enriched in the immunodeficiency cohort and absent in other cohorts and in the normal population, and whether it was predicted to be damaging. They were the main two variables, but many other things put into a smaller degree. So when they did that, they could then, for each given gene in the genome, attribute a posterior probability that that gene is associated with primary immune deficiency. And they came up with this list of genes along the bottom here and posterior probabilities that they're associated. The first thing we noticed was that in this top 20 or so genes, or a bit more than that, the blue ones are known genes. And that's heavily statistically enriched, and you can see they tend to be at the top end of the spectrum. When we first did this analysis, it was in about 2016-17, and actually one of these genes came up with a 100% probability of being associated with immune deficiency, and that was NFKB1. And because there were 14 cases of that among our 800 patients, 14 quite different mutations in different families. And so the algorithm flagged it up with a probability of 100%. It was then described in 2 families at about the same time. And we've subsequently been shown by our group and others to be the most common cause of primary immune deficiency in Europeans. So I should just say this algorithm works, and 5 new genes across this, and recently now 6 have been confirmed as causal as well. So among these green ones, we've now found 6 new causes of primary immune deficiency. So you don't need families to discover genes. You can actually, if you whole genome sequence large populations, you can find new genetic associations with disease. What you can also do by virtue of doing whole genome sequencing rather than exome sequencing is explore the non-coding space. And so, pardon me. Now, because we don't really know what's going on in most of the non-coding space, we decided we just look to see if we could prove the principle that it was possible to find defects in a non-coding space that might impact on disease. And we did that by looking under the lamppost, by essentially looking for genes that we knew caused primary immune deficiency when homozygous, looking for patients in our cohort that were heterozygous for a damaging mutation, and then seeing if on the other allele they were compound heterozygotes for a non-coding variant that could add together with a coding variant to create disease. And I'll just show you one example of this. And this is a patient here who had primary immune deficiency and a defect in ARPC1B. And this patient had a frameshift mutation, which would be predicted to cause disease when homozygous. And that's shown here. But on the other allele, she had a deletion taking out the promoter of the gene. And this is her extended family, so a large number of siblings. And she was actually deficient in ARPC1B, despite only having the frameshift variant on one allele. And you can see here that her mRNA was almost absent compared to her sibling, who was heterozygote. And the reason for that was shown with allele-specific expression assays that showed that the allele with the promoter deletion was completely incapable of making mRNA. So she's essentially a knockout due to a combination of a coding variant and a non-coding variant, which demonstrates the principle that by using whole genome sequencing, you can find non-coding variants that help drive disease and help drive monogenic disease. And we found other examples in DOC8 and LRBA as well, which I won't bore you with. Now, this is a difficult, we've looked at in the simplest way possible, really, to find these variants. It's going to be a huge underestimate of the number of non-coding variants that might have the same impact out there in unannotated parts of the genome, perhaps hitting super enhancers at distant sites, for example. But as the non-coding regulome is better understood, we can feed that into statistical frameworks like the Bevimed algorithm to help to pull out new genes from this sort of data resource. I'm going to show you 2 slides of things that were a bit of fun that just popped out, again, the benefit of this sort of sequencing approach. One of them was a patient shown here who our sequencing showed was homozygous at chromosome 1, so she had uniparental isodisomy. She'd inherited 2 copies of her mother's chromosome 1 rather than 1 from each parent, which meant that she was homozygous for everything on this chromosome. And she actually presented with recurrent purulent bacterial infections and was the first reported case of IL-6 receptor deficiency. Interestingly, she also had absolutely absent CRP, indicating that CRP in humans is completely dependent upon IL-6 receptor signaling. And one of the other things that came up that was amusing was a patient who, I don't want to say amusing, was interesting and as an anecdote at least, was a patient with X-linked IL-2 receptor gamma deficiency. Now, of course, this should cause X-linked SCID and be fatal in infancy in the absence of a bone marrow transplant. But this man was in his 40s with what looked like CVID clinically. And yet when we looked at his whole genome sequence reads here, he had, of course, this IL-2 receptor gamma stop variant here, but he also had other variation going on at the site. So we sorted his different cell subsets and sequenced them all independently. and found that while the stop variant was dominant in monocytes and granulocytes in B cells, he'd regenerated both his CD4-CD8 and NK cell lineages by a series of rescue mutations. So he had a mutation in a common NK T cell progenitor where cysteine had replaced the arginine, and that had recovered some of his NK cells and T cells. Additional serine replacing the arginine that had recovered the rest of his CD4 and CD8 T cells, and a leucine just in the NK cell lineage. So he'd been unlucky to have his original mutation, then spectacularly lucky to have corrective mutations, 3 of them, that had independently regenerated a lot of these lineages. But that was just a sort of anecdote. So we then went on to look at common variants, because this cohort's big enough to do a genome-wide association study. And of course, we've got whole genome sequencing data. So we have all of the SNPs, common SNPs available. So we did a standard GWAS, a minor allele frequency of greater than 5%. And we meta-analyzed that with an existing CVID primary GWAS done by Lee et al. An Icelandic study, which was published a couple of years before we did this. And so when we put these two together, we found a number of loci where we found significant associations between SNPs in the locus and primary immune deficiency. So of course, we're interested in finding out exactly what's going on at a common variant level, but we wondered if the identification of these common variant loci could lead us to novel monogenic causes of disease. So we prioritized these using an algorithm which Ollie Burren put together. I won't go into it in detail, but called his combined omnibus gene score. So the first thing he did was to try and enrich in a Bayesian way, the probability that a given locus was associated with disease by mining not only the primary immune deficiency genetic data set that I've just told you about, but looking at diseases with genetic relationships to primary immune deficiency. And so on the left is his graph looking at these relationships. So this is, again, the antibody deficiency subset of PID, which is 770 of our 800 or so patients. And this looks for enrichment of the genome-wide association hits in primary immune deficiency. And this is all variants, including subgenome 1, not just those few peaks that I showed you, against the genome-wide association study data from all of these diseases. And you can see strong associations with allergy and asthma, but also with Crohn's disease, lupus, to a less extent rheumatoid arthritis and UC, but not really type 2 diabetes or coronary artery disease as you'd expect. So when you put all that together, we could prioritize these loci as being a potential cause of monogenic disease. And I'm just going to talk about 2 of them, the PTPN2 and SOX1 loci. And the reason they're interesting, I think, is that they fall on a pathway that's already known to cause primary immune deficiency. So we know that patients with gain of function make mutations in STAT1, get excessive interferon-like signaling, and develop primary immune deficiency with both inflammatory disease and recurrent infections. So this is STAT1 phosphorylation, which is downstream of interferon. Now, SOX1, suppressor of cytokine signaling 1, directly inhibits this phosphorylation step. And so you'd expect that a deficiency in SOX1 would behave like gain of function STAT1 variants, and that's what the SOX1 deficient mouse phenotype suggests as well. Similarly, PTPN2 dephosphorylates STAT1. So again, its deficiency would be expected to increase phospho-STAT1 and look a bit like a gain of function STAT1 patient. And again, the mouse phenotype is consistent with that. So when we first looked at SOX1 haplo-insufficiency, we had only a single patient in our cohort. So it wouldn't have, because there's only a single patient, that wouldn't have flagged up by the Bevimad algorithm until a second patient occurred. And we found that patient had reduced SOX1 and increased phosphorylation of STAT1. We then contacted a number of colleagues in France and in, forgetting whether, and Germany and elsewhere. I found other patients, and you can see that in all of them, phospho-STAT1 was increased above baseline, and SOX1, as a result of SOX1 expression being halved roughly in this haploinsufficiency condition. So there's now been 9 cases of these reported. The interesting thing is it had been shown by a number of groups that ruxolitinib, a JAK inhibitor, was therapeutically useful in reducing some of the inflammatory disease granulomatous inflammatory disease seen in gain-of-function STAT1 variants. And as a result of us making these 2 diagnoses, our German colleagues and others have started to use in selected patients ruxolitinib in SOX1 haplo-insufficiency, and that's been effective. So it's a benefit of making these diagnoses. So we were led to this purely by the genome-wide association connection. The next and second one and final one of these cases is PTPN2. So this is the family we found, again, a single case of PTPN2 haploinsufficiency in the patient here, who presented at the age of 9 with severe deforming polyarthropathy, and then developed subsequently recurrent bacterial infections, was noted to have low B cells, and then developed interstitial lung disease and splenomegaly. Now, he was said not to have a family history of one of these many sporadic cases, but in fact, he wasn't a sporadic case, because when we chatted to his mum, she didn't have recurrent infections, but she did have 4 sorts of autoimmune disease, which we thought wasn't normal. And his brother had very severe atopy. So interestingly, while the patient had a stop variant on one allele, on the other allele, he had a SNP known to be associated in Japanese cohorts of rheumatoid arthritis. And his mum only had the stop and didn't have the common rheumatoid associated variant, and his brother had just the rheumatoid associated variant. So the question is, were these things combining to give this patient a more severe phenotype than the rest of his family? So we looked first at expression of PTPN2, and sure enough, the index case had a lower level of expression than his mother. And our phospho-STAT1 levels were substantially higher in the index case than his mother, suggesting that there was some sort of gene dose effect caused by this common variant. And to confirm, or at least get more evidence of this, and this is work done by Zinan Zhang before he returned to the NIH, and then to medical school, is that we looked for allele-specific expression to see what impact this G allele had on expression of PTPN2, and you can see that it almost completely wipes it out using samples from the brother. So in this first bit of the talk, I think what I've shown you is that by looking at a whole genome sequencing, a cohort of sporadic adult onset primary immune deficiency, we expanded our genotype-phenotype correlation. We found new genes in sporadic cases using this Bayesian statistical approach, started to explore the non-coding genome, and there's going to be a lot more in there, and found that we could use common variant loci to lead us to monogenic variants that help us understand rare disease. So, and this is by way of advertisement, if anyone wants to get involved in this sort of study. We've just commenced, after COVID-related delays, recruiting more of these patients as part of a Wellcome Trust Collaborative Award in League with Genomics England. And our plan is to double our number of sequenced immune deficiency patients, but also to recall 800 of them for detailed immune phenotyping and repertoire analysis, on the basis that we believe that by feeding these into novel analytic algorithms, we'll increase our ability to find new genes in here through statistical integration. So this is a big collaborative grant. So I'd like to now discuss some unpublished data that's the flip side of this. And there are many people involved in this study, but really it was conceived and driven by a PhD student, Will Ray, who's just finished his PhD. Who's also a clinical immunologist, and he's done about 3 bits of work like this during his PhD, so this is about a third of his PhD. And what he found was, from that cohort, or at least found it in that cohort I've just shown you, and then with collaborators adding more, 5 families with TRAF3 haplo-insufficiency. There are the mutations here. Interestingly, 2 of the families had exactly the same mutation. This is a Mexican family, this is a Cambridge family, so these are ethnically completely unrelated. And you can see that where there are multiple variants within a family, there's high penetrance, clinical penetrance of this disease. What he found was that this actually resulted in these variants, as you'd expect given that they're stops, resulted in reduced TRAF3 mRNA expression to about half normal. But interestingly, even more than half normal reduction in protein levels, and there's various reasons to imagine why that might occur, but it's very consistent observation. So these patients have half the normal dose of TRAF3, but they also have quite an interesting clinical syndrome. So of the 9 patients, they all had recurrent sinopulmonary and bacterial infections, 6 of them developing bronchiectasis, and some of them were quite young, so that this number would go up, I think, if they were all survived after 60. Most of them had splenomegaly and lymphadenopathy, widespread splenomegaly and lymphadenopathy. Widespread lymphadenopathy, you can't have widespread splenomegaly. Almost all of them had dysgammaglobulinemia. 1 had low immunoglobulins, the rest had hypergammaglobulinemia. And interestingly, that 6 of the 9 had autoimmune disease, and in 5 of those cases, it was Sjogren's syndrome or a related sickle-like syndrome. So they seem to have clinical features of B-cell hyperproliferation, at least in terms of those 3 things. So this is just an example of the spleen. They did a splenectomy on one of these patients, and they had huge numbers of very large reactive germinal centers, shown here. The same patient at MRI had changes consistent with Sjogren's syndrome in their parotid gland. And another patient had a lower lobectomy, and you can see bronchiectasis, but also tertiary lymphoid follicles within the lungs. So consistent with that clinical phenotype, these patients had consistent changes of their immune phenotype by flow cytometry. So they had reduced CD4 T cells, shown here. If you activated their T cells in vitro, they had reduced IL-17 interferon gamma production, which is relatively specific. They had normal IL-13, for example. Reduced switched memory in their B cells, but an increase in these autoimmune-associated CD21 low B cells that you'd be familiar with in rheumatoid and lupus, and increased plasmablast differentiation in vitro. So in contradistinction to their memory cell reduction, suggesting an alteration in differentiation direction between plasma cells and B cell memory. Now, just to remind you, this is a rather old review, but an old diagram, but TRAF3 is known to be an inhibitor of the non-canonical NF-kappa B pathway, which is driven by increased NIC and dependent upon phosphorylation of P100. So mouse studies and in vitro human studies suggest we'll be removing the brakes from this non-canonical NF-kappa B pathway. TRAF3 deficiency in mice is very complex. There's evidence for dysregulation in cell-specific knockouts, T cells, B cells, and myeloid cells. And in different contexts, these mice get predisposition to both autoimmunity and infection. So broadly consistent with what we see in humans, but hard to marry up exactly, because the humans, of course, have haplo-insufficiency of the whole organism. These mice have complete deficiency, but only in individual cell subsets. So you can't quite marry the two together. But as we'd expect from those early studies, reduced TRAF3 in humans leads to this increased NF-kappa B2 non-canonical NF-kappa B pathway activation. So there's increased NIC, increased phosphorylated P100. So the NIC protein expression is shown here across a range of different families. Phosphorylated P100 is enhanced. And in fact, the overall production of both P100 and P52, which is required for nuclear localization and the impact of this pathway, are enhanced. We see gene enrichment in naive B cells for genes known to be involved in that pathway, and some of those are shown over on the right here. But we see more than that. We see that has a knock-on effect on the B cell metabolism. So the B cells in these patients have enhanced oxygen consumption rate and OXPHOS. And if we transfect them, and this is BLCL cells, if we transfect them with TRAF3, that can be reversed, shown in blue. Consistent with that, they also have reduced mitochondrial complex 4 activity. And this is an electron micrograph of a normal BLCL. But from our TRAF3 haplo-insufficient patients, you can see markedly abnormal mitochondrial morphology, which again we can correct by transducing back in TRAF3 and rescue that. So we thought that was interesting. It's a new monogenic cause of primary immune deficiency, but an interesting one with this syndrome that has quite unusual features for the sort of primary immunodeficiency we've been talking about. We wondered if the fact that these features seemed unusual and sort of relatively specific could inform us about common variants and a common or polygenic disease. So we decided to do a focused genetic assessment, looking just at syndromes that are suggested by these the features of this monogenic syndrome: Sjögren's, lupus, and celiac disease, immunoglobulin levels, bacterial infection. And even though we didn't see B-cell malignancy in any of our patients, interestingly, 5 to 10% of myelomas and diffuse large cell B-cell lymphomas are known to have somatic mutations in TRAF3. And of course, we know that 10% of Sjögren's syndrome patients get a B-cell lymphoma. So we threw into the mix multiple myeloma and B-cell lymphoma. And the rationale behind doing this is by focusing on these specific traits and on one disease, we can reduce the statistical bar for significance and make some of these genetic questions tractable with existing cohorts. So when we looked for these common variants, and a slightly busy slide for which I apologize, we found firstly that there is a significant statistical association with both myeloma and non-Hodgkin's lymphoma, which reaches significance. And that's the first evidence that germline variation in TRAF3 can predispose to these B-cell malignancies. And you can see here that the EQTL effect size, in other words, for a given SNP, the impact that it has on TRAF3 expression is inversely proportional to the effect size of that SNP on risk. So in other words, the bigger impact on TRAF3 expression, the bigger impact on risk. So this is consistent with TRAF3 playing a direct role in driving risk of malignancy. And interestingly, from another published data set, if you just divide patients with myeloma into the half with the highest 50% expression of TRAF3 and the bottom 50% expression, you can see quite a difference in clinical outcome, again, suggesting that TRAF3 is playing a role in malignancy. We didn't find an association with bacterial infections. And this is, I must admit, been our group's experience in a number of different settings, largely because the bacterial infection GWASs have two problems that I think make them less hard to interpret. The first is that they're usually GWAS looking at individual bacterial infections, so pneumonia once versus not getting pneumonia. And in other words, so sort of sporadic infection that isn't associated with a genetic cause may be crueling the pitch. And if instead there was a GWAS of recurrent infections, I think that would be far more tractable. The other reason is that in most of these GWASs, lots and lots of different bacterial infections are thrown in together, which I think is likely to create noise. So we didn't see an association there. We did see an association with lupus, strong association. We didn't see an association with Sjögren's or celiac disease, but the number of cases that we had to analyze were tiny. And I think this could well be related to power. So I wouldn't call those definite negatives, but they certainly weren't positives in our example. And then an extremely strong association with immunoglobulin level. No cases and controls here because we did a genetic analysis with immunoglobulin as a continuous variable rather than dividing into two groups. So what we've done is to use information from rare variants shown here to understand how this gene impacts lupus. And this, I should say, we did this study with Tim Vise, who many of you know very well, lupus geneticist at King's in London. Globulin level, which had been previously reported by a number of groups who've done large immunoglobulin GWASs, and myeloma and non-Hodgkin's lymphoma. But we wondered about what's going on in the middle. And I think, and this is sort of obvious perhaps, but a lot of us have overlooked this, that genetics has really been running down two tracks for the last decade or two. The monogenic track, which is primarily familial disease in young patients, although as I've shown, it's now tractable to look at sporadic disease in adult patients, and the common variant track. So these variants are incredibly rare, less than 1 in 100,000, or you don't consider them likely to be pathogenic. And these must be very common, and most GWASs have a minor allele frequency cutoff of 5%. Some of the big ones now 1%. But in between 5% and almost nothing are a lot of variants at intermediate frequencies, and we have ignored them largely. We don't know what they contribute to disease, and that's just because we haven't had the tools and large enough cohorts to investigate that space. So we wondered, using our targeted approach, whether we could do that. Just as an example of what's in that space or what might be there and might be there in great numbers. This is the slide I showed you earlier of our primary immune deficiency GWAS with a 5% cutoff. If we drop that cutoff to 0.05%, we get a lot more noise, but we see this. And this is TACI, a member of the TNF receptors superfamily. And TACI was originally described as a monogenic cause of primary immune deficiency, but subsequently found not to be monogenic. It's enriched, so 6.8% of our 800 patients have a damaging variant in TACI, but 90% of patients with these variants are perfectly healthy. So this contributes significantly to your likelihood of getting an immune deficiency, but it isn't a monogenic cause, and it sits in this very hard to measure intermediate frequency range. So when we looked in the same range for TRAF3, we found a variant, and we looked in the literature, and it had been reported. And it had been reported back in 2010 as in a single case of herpes simplex encephalitis as a monogenic cause of primary immune deficiency, or monogenic cause of or predisposition to herpes simplex encephalitis, and was shown to have reduced expression, and some functional studies have been done in fibroblasts that were all consistent with that. But that was published before large genome databases or whole genome sequence databases were available. And it's now clear that this variant was it's found in 1 in 200 to 250 of the European population. So it's clearly not a monogenic cause of anything, because it's really quite common and in this intermediate frequency range. And in fact, when we looked in the UK Biobank, we couldn't find any cases in the 2,500 patients who had this variant, any cases of herpes simplex encephalitis, despite the fact that they had normal reported herpes simplex infections, and their herpes simplex IV titer or positivity was equivalent between the two variants. So they're not more likely to get herpes, and they wouldn't appear to have at least a substantially increased risk of herpes simplex encephalitis. And obviously, therefore, it's not a monogenic association. But what is it? So we again did our targeted GWAS approach in the UK Biobank initially, and then where we could find them in replication cohorts. And we found similarly, as we've done with the more common variants, in association, a germline association with risk of myeloma and non-Hodgkin's lymphoma, we now came up with a significant association with bacterial infections, but we couldn't get a decent replication cohort for that. We again found an association with lupus, which replicated, and with immunoglobulin level. And so we then proceeded to get, we've now got about 20 patients, but these are the first 5 patients. I haven't had time to make the slide. These are not patients, these are volunteers that are part of the NIHR bioresource, which is set up in Cambridge, but is now a national study with about, I'm not sure, but I think 60,000 volunteers who've agreed to be recalled for studies and have provided DNA. So they've all been genotyped. We've got serum and DNA in the freezer. And then what we did was to look for patients who were heterozygous for this variant, and then got their bloods along with matched controls. And what we found was they did have reduced TRAF3 expression, and the increase in numbers have got all these p-values to quite markedly significant. So they were almost, they had reduced TRAF3 protein, increased phosphorylate, increased P100, increased P52, and a quite marked peripheral phenotype. So I won't show you all of it, but for example, they had marginal zone-like cells in the blood were reduced, plasma cells were increased. So even though these were members of the normal population with no history of disease of any sort, they had a reduction in TRAF3 and an associated immune phenotype that was easily measurable. So I think that tells us that we now have the tools to start working out what's going on in this gap between rare variants and common variants. And I think it's going to be increasingly possible to study these things all together as we discover either common variants associated with disease or rare variants, as our cohorts get bigger and can be dug into in more detail. And we can start to understand in a global sense how a variant or a pathway contributes to human disease, linking rare and common disease with increasing frequency. So I think I've mentioned most of the people involved in the work as I've gone along. I have on different slides. So I thought I'd finish with this one, which is just a summary, which I've shown you an example of where common variant associations in primary immune deficiency and autoimmune disease can drive monogenic PID gene discovery. And that can identify pathways that drive disease, and in the case of SOX1, suggest specific therapies that can be of benefit in patients. But conversely, we can use monogenic immunodeficiency clinical phenotypes to drive autoimmune gene discovery in the large genotyped cohorts, which increasingly have linked clinical data that are now available. And the example was TRAF3 Haplo-insufficiency to an understanding of TRAF3's role in common disease, not just in using common variants, but exploring the role of intermediate frequency variants, which could have just as big or bigger impact on common disease than the common variants we've been studying so far. So I'm sorry about the London Underground specific logo there. This is obviously designed for a British audience, but I'm sure some of you get that. So we're beginning to understand this group of immunodeficiency patients, and I think increasingly we'll understand how dysregulation here relates to the dysregulation of rheumatoid arthritis and lupus, and define ways of describing in a much more nuanced but accurate way how different genetic variants come together to drive disease. So Thomas, that's all I have to say. Thank you, and I'll de-share if I can. Can't.
Thanks very much, Ken. I have a question first, while we wait for others to type in some questions. I wonder if you could synthesize what you said about the TRAF3 variant in lupus. It seemed to me that you indicated that TRAF3 is involved in several separate biological processes. So if you put it together and you say what cell types are affected and how does that variant inform us about lupus pathogenesis and what cell types and pathways are important there?
Yeah, so I think yes, I spared you that just because I've only got an hour, but so we're digging into that now. So, and one of the interesting things is that the patients, have this B-cell hyperactivity syndrome, but there are reduced switched memory B cells, but enhanced plasma cell production. If we look in more detail, we see a substantial increase in circulating T follicular helper cells. Mm-hmm. And we also see where we've got histology enhanced germinal center responses. So I think that it's likely that this dysregulation, which will involve both T cells as well as B cells, is driving an enhanced germinal center response, but in a way that we don't fully understand is also impacting on differentiation in that. So those enhanced germinal centers aren't making more memory cells, they are tending to make more plasma cells. And consistent with that, we see changes in the B cell repertoire and somatic mutation that would fit with that. So I think it's not going to involve one cell type. It's going to be a T cell, B cell thing at least. A number of people have also reported substantial myeloid changes in TRAF3 haplo-insufficiency, in TRAF3 deficiency in mice. We don't see gross changes in the myeloid cell populations in these patients, with a possible, or in either the patients or the controls with that intermediate variant. Possible exception of PDCs, which we're repeating those studies to see if that's real. They seem to be increased. But I think it's about germinal centers. Primarily the lupus angle will be about germinal centers, and it may well be about T follicular helper cells and the control of differentiation with and germinal center output.
Any potential connection with type 1 interferon production from that variant?
That's a good question, and one that I can't ask directly. I mean, certainly toll-like receptors are increased and are known to be, TLR signaling obviously can go through this non-canonical pathway, and there are feedback loops. So we do see increased TLR9 and TLR7. Interferon I haven't specifically looked at, but it's something we should look at, obviously, because it's particularly given the lupus angle. Yeah.
So we have a question from Eric Allensbach. Wonderful presentation. Many of the initial cases you presented, SOX1 haplo, PTBN2 haplo, TRAF3 haplo, with novel PID were coding variants. What is the percentage of improved solve rate with whole genome sequences over WES in your cohort?
Yeah, so that's a very good question. we had a very long discussion with reviewers about that. We had an 18-month discussion with reviewers actually to get that Nature paper published. The answer is not high at the moment. So almost all of, well, pretty well all, Bevimab was really only successful in picking up coding variants and was designed for that. So we could have used that Bayesian approach. We could have just done that with whole exome sequencing. Of course, we could have done the GWAS with standard GWAS SNP analysis. So what do we gain from whole genome sequencing? Well, we believe that what we will gain, or we gain those few examples I showed you of non-coding variants that we'd managed to find. We get a much better hit rate at detecting deletions. Deletions are hard to detect from whole exome sequencing, and big deletions can be important. So we found quite a few of those, which I didn't talk about. But I think the main benefit of doing whole genome sequencing in these cohorts is actually for the future, because I think increasingly, as we develop a better understanding of the non-coding space, our hit rate and of intermediate variants that are usually non-coding, not always, but usually non-coding and can impact on disease. I think that the benefit is in the future. And if we develop big cohorts that are whole exome sequenced, we'll have to resequence them at some point if we're going to make that jump. And it's probably better to start now, particularly since the price gap between the two is getting a lot lower. But it's a fair point that you could have heard almost the same talk from whole exome sequencing, but it would have been less future-proofed.
So a question from Gordon Starkebaum. Are there any somatic mutations that contribute to PID?
No, that's a very good question. And there's been a couple of examples of those published, but the answer is I think that's a field ripe for exploration. Because, I mean, I think the example I showed of where we'd actually effectively found somatic mutations from the from whole genome sequencing of that patient with the reversion mutants, that shows that there can be profound impacts of somatic variation or immune function, albeit that was an unusual case. But the answer is no one knows the extent to which somatic mutation in primary immune deficiency, sporadic primary immune deficiency, is important. I would be shocked if it wasn't important, and it needs to be looked at systematically.
So Gordon also is asking, how do you account for the late onset of sporadic PID?
Yes, so this is where immunologists hand wave, of course, because immunology is complicated, is the answer. That I think there are going to be sort of second hit events, be they somatic mutation, but more likely things like infection and exposure, that reveal these diseases with age. And I think some of the impacts on the immune system will worsen with age. For example, telomere shortening, and so, you know, there are problems that arise with long-term immune dysregulation that just make these diseases more likely to occur with age, or present with age, as is the case with, you know, normal rheumatoid arthritis, for example. So I think a lot of the factors that drive late onset of common diseases are going to be equally important in these monogenic diseases that don't have a sort of severe early phenotype. But we can hand wave about what they are.
Yeah. So a question from Eric Butz. To what degree is the identification of these variants actionable for treatment of patients' disease? Yeah,
so increasingly there are examples, like the one I gave with SOX1, where there are tools available now that allow us to treat those diseases. In the case of TRAF3, for example, I mean, you would be tempted to use rituximab in a patient who had nasty inflammatory disease because we have clear evidence that B-cell dysregulation, either primary or secondary, underlies that. So I think increasingly we'll be able to make more intelligent choices about the use of existing therapies. Of course, the great hope is that the gene therapy will become simple enough for us to actually get to the heart of the problem, but that's a fair way off. But it's also worth emphasizing that genetic counseling is a really important component of getting a diagnosis in these patients. Particularly given that they're often young families who don't know what the future holds. So I think there's benefits beyond straight out therapy. And that can involve early identification of disease in at-risk relatives, or just counselling for future family planning and so on.
So Ken, do you think in the coming years we're going to whole genome sequence every rheumatological patient?
I think we're going to whole genome sequence everybody at birth before very long, because this is, all the there's more than just the immune system out there apparently, although I try to ignore it. And I think increasingly it's going to look like a pretty small investment in an expensive lifespan, and we're going to be doing it for everybody.
Yeah. There's a question from Keith Elkon. Did you say loss of function variants of NF-kappa B also cause autoimmunity? If so, do you know how it works at a cellular level?
So no is the answer to that, except that it's complicated because obviously it's impacting all immune cells. But I think that's the fascinating. I mean, I'm not going to answer your question, and this is not a way of getting out of it. But I mean, the fascinating thing here is that turning the same pathway up or down in some of these contexts, either one can give you inflammatory disease and infection. And I think some of us might remember, and I think I may have even been a victim of this in my youth, being given this seesaw slide in your undergraduate lectures of, too much immunity is autoimmunity, too little is infection. Mm-hmm. And of course, we all know that that's nonsense. Abnormal immunity is abnormal immunity, and generally increased risk of infection goes hand in hand with increased risk of autoimmunity. And so I think it's fascinating to think about things like NF-kB1, where, as you say, you turn it on or you turn it off. Either way, you end up with a range of both inflammatory and infectious complications.
Mm-hmm. So do you think also in the area of immuno-oncology, like checkpoint inhibitor use, that the variation you see in efficacy, the ability of the immune system to eradicate tumor, is related to all of this biology in a similar way?
Yes, I mean, it would be hard to imagine that it isn't. And I think, there's early hints, certainly there's extreme cases where it's obvious, but there's early hints even from GWAS that that's starting to be the case. What would be fascinating is to develop a more systematic way of analyzing someone's overall immune risk. And I think, while that's, genetic risk scores, for example, are becoming tractable in simple diseases, I say that carefully, but, I wonder if increasingly we're going to be able to make more generalized comments about immune fit using that sort of approach. Mm-hmm. But that probably is a little bit far off for being practical.
And would you extend that also to the immune-related adverse events that patients often experience?
Absolutely, yeah. And I suppose what we don't yet know is how much of this is going to be genetic and how much is going to be, bad luck and sporadic in different ways. And but I think, and that's where digging out both understanding the non-coding space and these intermediate frequency variants is going to be important.
Yeah. We have a new question from Sean Jackson here. Have you looked whether additional genetic variants impact the phenotype of specific PID syndromes? For example, in subjects with pathogenic gain of function mutations in STAT1, do additional genetic variants impact whether individual patients present with chronic fungal infection versus autoimmunity? Yeah, the patient numbers too low.
So it's a good question, and I think that in the past patient numbers have been too low, but I think they're not going to be too low. So and what we haven't yet done, and it's one of the projects that is sort of happening now, is, for example, we've got 6.8% of these 800 or so patients. We've probably now got, we've certainly got over 1,000 patients. So that's 70 or 80. We're going to have people with a TACHI variant who actually have, many of whom will have other defects. We know that. So we'll be able to systematically then say, well, how does TACHI impact on those variants to drive disease? And I think the numbers are starting to be tractable for some of those combinations. And obviously there'll always be combinations that are too rare to see, but I think we'll be able to prove principle that we can understand those interactions anytime now. I think we're starting to get to the numbers.
Yeah. Okay, I think we are up to the top of the hour, and I've exhausted all the questions here typed in. So I'd like to thank you very much, Ken, for a wonderful seminar, and hope to talk to you soon again.
I hope so, and perhaps in person next time.
Yes.
Okay.
Thanks. Bye-bye.
Great. Thanks. Bye-bye.